So when I first got the title for this talk, it was LAM Research Worldwide, which I thought was quite a lot to cover in a 20-minute slot, in a really good way. So what I want to do instead is try to explain to you how the work we do in the lab, how basic science results in new therapies for LAM. And what I want to do is talk about clinical trials that are ongoing for new therapies, and also the work that we do in LAM, in our research lab in Nottingham, in Simon Johnson's group. So as Kelly said, I'm a research scientist. I work at the bench, and I'm going to have to go through the science bit a little so that you understand what we do. You need to know that LAM is a disease caused by having a cell in the lung that really shouldn't be there, and that cell has an error in the DNA, and because of that, it can't make a protein called tuberin. We know a lot about what tuberin does in the cell. It regulates other proteins, and those proteins then regulate what the cell does. If tuberin isn't working, that means the proteins it regulates don't work properly, and that means the cell doesn't work properly. But because we know so much about what tuberin is doing in the cell and these pathways, we can work out some new therapies because we understand these pathways so well. We understand the science of these pathways well. And the first drug that came out of this understanding was rapamycin itself, which is an excellent inhibitor of this pathway. As many as you will know, rapamycin is now a therapy for LAM. And it's a really good therapy. It can stabilize LAM, but it does have side effects. The other problem with rapamycin is you have to keep taking it continuously. It seems that if you stop taking rapamycin, the disease progresses again. So we need an additional therapy. What we need to do is actually wipe out the LAM cells. We need to kill them. We don't want them just put on hold for the time that you take rapamycin. We need something else to take as well so that we can eradicate those cells entirely, and that is a cure. That's what we're aiming for. So LAM has some advantages in that many of the processes that occur in LAM also occur in other diseases. For example, in pulmonary fibrosis and in cancer. It doesn't mean that LAM is cancer necessarily. It just means that the same processes happen. And we can benefit from all the research that's already gone on in cancer, and all the pathways that have been defined in cancer, and all the therapies that have been identified for cancer, and we can see if those will work for LAM, which means we can make really quick progress. So this is a list of some of the clinical trials that are ongoing at the moment with new therapies for LAM. And if you want to look at this, actually, you can look at clinicaltrials.gov. And if you go there and put LAM or lymphangial leiomyomatosis in there, you will see a list of all the ongoing trials, some that have closed and some that are still recruiting. And what they'll be measuring is the output. The first one I'm going to talk about is this one here, but only very briefly. It's a trial of a drug that's been used in pulmonary fibrosis before, and it's been used in some lung cancers. And the reason it's being used in LAM is because researchers have found that the target of that drug, which is called PDGF receptor beta, which you don't really need to know, is present and active in LAM-type lesions, which means that this might be one of the causative features of LAM. This might be one of the pathways that are causing LAM cells to proliferate. So this is why this drug is being trialled in LAM. The next trial has the acronym COLA. Most trials have a cool acronym. I don't know why the first one doesn't, but all the other ones do. So I'll explain why this COLA trial is ongoing. We know that estrogen must be important in LAM, but we don't exactly know why. We know it's important because only women get LAM, so it seems obvious that it's hormonal somehow. But the actual role of estrogen hasn't been shown. But what has been shown is that if you put estrogen on a cell that has exactly the same kind of mutation that the LAM cells have, they produce something called COX-2. COX-2 then causes release of something called prostaglandin, and prostaglandin makes cells proliferate. So you can see that what can happen now is you have the estrogen, it makes your cells produce prostaglandin, that causes the cell to proliferate, you have more of this, and off you go. You get more growth because you're producing prostaglandin. COX-2 is found in LAM lesions. It is expressed in LAM lesions. And women with LAM have higher levels of this prostaglandin in their blood. So the bench science is backed up by patient data. So what we really need is to inhibit this. What we need to do is stop this working. If we can stop this working, we don't get prostaglandin, and then we don't get this excessive cell proliferation. So that is what this trial is all about. This trial tests an inhibitor, celecoxib of COX-2. And that's why the COLA trial is ongoing. This is what this trial is designed to do. And I thought this one was maybe quite interesting because there is another very well-known inhibitor of COX-2, which is also therefore a potential LAM therapy, and that's aspirin. So aspirin itself, in combination with rapamycin, actually might turn out to be quite beneficial. And the results of this trial will inform us as to whether actually it's a good therapy for LAM. So that's the COLA trial. The next trial is called LAMP1 and is a trial of a drug called imatinib, which is also called Gleevec. This has been an extremely important drug for treating cancers. It hits a few different proteins. This one's called ABL, this one's called c-KIT, and here, I don't know if you can see it very well, it's quite dark, it's the PDGF receptor again, the one we were talking about with the first trial. And it really has transformed the treatment of some cancers. It's a very powerful drug. The reason it's being used in LAM. So, these are its targets, and Dr. Jean Damentio has shown that this drug will completely block the growth of LAM cells through the initiation of targeted cell death. And the important word there is death. That's what we're looking for. We're looking for a drug that can actually kill the cells, not just stabilize them. So that is a LAMP1 trial. The last one I'm going to talk about has the acronym SLAM2. The target of SLAM2 is a protein called SERT kinase, also elevated in lots of cancers. So there are lots of drugs that can treat SERT kinase, and this is one of them. The reason this is interesting is the reason that LAM cells seem to have more of this is because they have the TSC2 mutation. So they have the TSC2 mutation, so they make this kinase, and that means actually that all the pathways that this kinase is involved with, this one causes blood vessels to grow, this one causes proteases to be made that can degrade tissue, this one anchors cells to matrix. All these pathways can effectively be working too hard because the cell is making too much of this protein. So the inhibitor hits this protein here to stop those changes occurring. So those aren't all the trials that are ongoing at the moment. These are the ones that I thought actually would allow you to see how bench research translates into therapies by using information that comes from other diseases. So really quick progress can be made. Is what we've identified this pathway, there's already a drug for that, it's fine. We've identified a different pathway, there's a whole plethora of drugs. And then these can immediately go into clinical trials with LAM patients. And the output of these trials will be stabilization of lung function or VEGF-D levels, or even how big or small your AMLs get. So if you're interested in potential trials, look at clinicaltrials.gov and you'll find some information there about other trials ongoing, like simvastatin that you might have heard of. But that's all there. So I'm now just going to talk about the research we do in our lab. We've always had the same aim, really, in the research that we do, and that is to understand why the lungs get damaged so badly in LAM and how we can stop it. And we have a model of LAM that was generated in our lab, and now I think is quite well accepted, where it's not just the LAM cells that are a problem. We think the LAM cells come into the lung, and then other cell types come in alongside them, these green cells and these orange ones. And some of the effects that you see in LAM are not because of the red cells, the LAM cells. Actually, the other cells play a role as well. So that's what we're interested in. So, what we want to know, is the lung destroyed by protein-damaging enzymes? This seems to be the kind of common consensus that it must be an enzyme, then too much of it gets produced and it starts breaking down the structure of the lung. So proteins like collagen and elastin are what form the framework of your lungs. If they get broken down, then you can get these cysts formed. So that's the first thing we're looking at. The second thing is there some just change in the structure of the lung that somehow makes it more fragile? So it's not really to do with proteases, it's to do with some other structural change. Or could it be the two things together? So what we look at is which type of cells are causing these proteins to be produced or these lung changes to occur. And the other question is, how do they get there? How do they get into the lung? Why do they associate with the LAM cells? Because these are the processes that we can target with different drugs. So I'm going to go through two of the ongoing projects in the lab. The first one is to do with this protein cathepsin K. Now, cathepsin K is really, really good at breaking down collagen and elastin, just the proteins that are forming the structure of your lung. And it's normally made by fibroblast cells, but in a sort of regulated way. They don't go mad, they produce a little bit just to do their job, to remodel collagen and elastin, but it's all very regulated. We know that in LAM, we have quite a lot of cathepsin K in exactly the LAM nodules. We can see where proteins are because we can stain them brown. This is a normal lung. This is what we call a LAM nodule, where we get this community of LAM cells and other cell types coming in. And when we use our brown stain to find cathepsin K, we find there's quite a lot of it there. Now, there is one thing about cathepsin K which is a bit different, and that is it really likes acidic conditions. We look at how active it is, how good it is at breaking things down under not very active conditions. And if you just look at this column here, under not acidic conditions, and there's a bit of activity, but not loads. But if we make the conditions acidic, the bar gets much higher. There's much, much more activity of this protein, cathepsin K, when its surroundings are acidic. But how would it get into an acidic environment? And the problem is the LAM cells themselves. LAM cells are very good at making their environment acidic. The cells that have the TSE2 mutation that don't make tuberin properly also cause their environment to become acidic. And this is us measuring the acidity. Conversely, if it goes down, it's actually more acidic because it's a pH measurement. So as we grow them, what we find is the medium they're growing in gets more and more and more acidic over time. So what we think is happening in the lung, this is our model. We have a LAM cell and it sends out a signal. Those green cells are our fibroblasts, making a little bit of cathepsin K in a completely sensible way. When the LAM cell sends out its signal, the fibroblasts come running. We're working on what this signal might be, and we have a few ideas, but that's one of the things we want to identify. And they all huddle around the LAM cells. Now, the problem is, at that point, the LAM cells start to make their environment acidic, which we know they do. And then the cathepsin K that the fibroblasts are making is suddenly much, much more active than it should be. And we think this is one of the things that's breaking down the lung structure. You've got a combination of two unfortunate things, fibroblasts making an enzyme and cells making exactly the right environment for that enzyme to work really, really well. So just by chance, those two things come together, and what you get then is a massive amount of activity, which is really destructive. So we have to start turning this back. And the first thing is cathepsin K itself is what we call a druggable target. There are inhibitors of that because it's involved in osteoporosis. So this is another example of where research in another field actually is really beneficial to us in LAM. So we can get rid of that. What Arrow has been doing as part of a PhD is working out exactly which proteins it is that are making the environment around the LAM cell acidic. And a lot of those are druggable. So we can get rid of that. And the final thing we want to know is, what's this signal that's attracting the fibroblast to the LAM cell? Because if we can get rid of that, everything would go back to normal. So this is the outline of our cathepsin K project, which actually, our preliminary data were used to get some more grant funding. So we've got more grant funding to work on cathepsin K, and also there's a paper imminently coming out. We've just been correcting the proofs this weekend. So this work is just about to be published. So that's one example of where we think proteases might be involved in breaking the lung down, which is sort of the standard idea. but we're looking at a different protease to most people. The other idea is a little bit different, and that is maybe something happens to the structure of the lung which isn't to do with proteases, but which makes it a little bit more fragile. So I'm going to talk a tiny bit about the structure of the lung. This is an alveolus where gas exchange occurs, and there are two types of cells that make up this balloon, if you like. There's some really, really thin walls that make up most of the wall, and they're type 1 cells. And there are these little fat ones here. Now, these are called the type 2 alveolar cells, and their job is to make type 1 alveolar cells. So if this one gets damaged, this one will divide and give you a new one. If these get damaged, the type 2 cells divide and they make more type 1 cells. So that's how your lung, if it gets damaged, makes more alveoli. And we know that the type 2 cells love a protein called FGF7. If they see FGF7, they will migrate towards it. And they'll proliferate in response to it. They really like this protein. Now, in LAM, what we find is this is an area of LAM here, in a relatively kind of normal bit of lung. It's absolutely plastered with type 2 alveolar epithelial cells. They just go and surround a LAM nodule. There is something in the LAM nodule that those cells really like. So here they are, they're around here. Again, I've stained them brown using a brown stain attached to an antibody. So what you get is something like this. You get your LAM nodule, like I showed before, and ... you get a layer of alveolar epithelial cells. Now, this is one of the things, one of our ongoing projects is to build LAM in a dish. So this is one of the things that we're trying to build in the lab. We put together fibroblasts and alveolar epithelial cells and LAM cells, and we actually build this so we can do experiments on it. But why do these cells migrate here so quickly? Well, what we think it is because LAM cells make the fibroblasts make FGF7. So if we grow LAFs, these are just the fibroblasts on their own. They make a little bit of it. But if you grow them with LAM cells, suddenly they make tons of it. The LAM cells themselves, which are here, are not making any. What they're doing is they're telling the fibroblasts to make a lot more of it. So in LAM, what we think is happening is we have this little community of LAM cells and fibroblasts forms, as well as the cathepsin K issue that I've already talked about. The fibroblasts start making FGF7. And then the alveolar epithelial cells come in from the surrounding area. And we can sort of do that in a dish. We can make models of type 2 alveolar epithelial cells come through this sieve to meet the fibroblasts, and then we can count how many come through. And compared to having no fibroblasts in the bottom, lots and lots of cells come through if we have fibroblasts in there. So they really do migrate. And what we think the problem with this is, the type 2 alveolar epithelial cells aren't doing anything bad where they end up, but they should be somewhere else. What we think is happening is by bringing them in from all the surrounding areas around the LAM nodule, those areas now don't have enough of them. So what happens if your type 1 cells around there start wearing out and needing replacing is there aren't enough type 2 cells to make them. So by drawing them all into one area, you've left the surrounding area now more fragile. So it's not to do with proteases, it's really to do with repair. What we think is that LAM might cause the lung around a nodule, it can't repair itself as well. So that is our hypothesis, that loss of type 2 alveolar epithelial cells around a nodule makes that area more fragile, so you get these cysts forming. So these are not the only projects that are ongoing in our lab. There are some others, but I think these are the ones that are probably most complete at the moment, and they really highlight our hypotheses the best. So I'm just going to conclude to say there are lots of really promising drugs in clinical trials that have had real benefits in other diseases, and there is really good science behind them for using them in LAM, and they're already in clinical trials, so things are moving very, very quickly. The amount of research that's coming out on LAM is increasing year on year. If you just look for how many papers people produce about a particular subject, you'll get a graph that normally looks like this. For LAM, the number of papers is just going up and up. So much more research is coming out on LAM. the more research is done, the more research begins on the areas that have opened up from that research. So it just snowballs. Our LAM research group is expanding. So because of our really good preliminary data, we've made grant applications that have brought in more money. So we now have more people working on LAM. And what we're working really hard on is to understand how these cells in a LAM nodule talk to each other and what the consequences of that are and how that results in destruction of the lung. And what we're particularly looking for is what everyone is looking for, which is a target, a druggable target, something that's already been identified for a different disease for which there is a drug already available and that is ready to go to clinical trials. So that's the aim of our research. And obviously, just a quick acknowledgement slide. Obviously, thank you very much to all the patients, anyone who has donated any tissue at all, because this is incredibly valuable for our research. LAM Action, as Kelly said, have paid my salary for 15 years, so thank you very much for that. I've presented some of Aru's work here. Quite soon, hopefully, that will be Dr. Aru, because she submitted her PhD thesis yesterday. And her PhD has been funded by LAM Action, and she's been working as a postdoc under LAM Action as well. And we now have 2 new members of the lab who are contributing to our LAM research. And that's it. Does anyone have any questions at all? Lucy?
I just wondered if all that lovely LAM research you talked about is being done elsewhere.
No, you know, the good thing about LAM is everyone has their niche and there tends not to be a massive amount of overlap. In the UK, we're the only LAM group. In America, there are quite a lot of LAM groups, but there is no one who is overlapping with exactly what we're doing. So, I mean, it's a good thing, actually. But yeah, so there is interest in what we do. There's a lot of interest, but we're not competing with anyone particularly.
What I meant was the clinical trial is now in process.
Yeah.
Can anyone here in the UK sign up?
No. Some of them are local. So one is based in Milan, others are based in various hospitals, and some are recruited. Sam will have a better idea if there's any that you could contribute to from here, but I wouldn't have thought so. They tend to be recruited from local populations.
Which is quite disappointing.
Yeah, no, obviously.
Enthusiastic group.
Yeah, and it's disappointing on both sides because obviously they want the patients to get the power for their study and the patients want to be involved. But, yeah.
Okay, thank you.
Hi, is the work that's being done on alveoli and the mTOR pathway also applicable to the angiomyolipoma?
Oh, no, actually. I can talk about the angiomyolipoma next year if you like. So women with LAM, a lot of them will also get these benign fatty tumours called angiomyolipoma. We think something different is happening in the angiomyolipoma. So that because the LAM cells, instead of ending up in the lungs, they end up in the kidney, the actual environment around them is quite different. So they don't have the opportunity to recruit fibroblasts, for example. What we think happens is they sense the environment around them there, and what they do is they develop along a particular pathway where they turn into fat cells. And so that is, I think, that is simply because of the environment that they find themselves in. So you don't get cysts or any of those things that you see in the lung, because actually the cell has decided, because of where it is, to do something different. And we think it develops as fat, and then it sends out signals, and that causes blood vessels to form. So we think a different process is going on. But we're studying that as well, actually. Okay, shall I hand over to Simon?